All right, we are getting to the bottom of a question today, and that is if you want a natural or biotope tank, what should you spark your ecosystem with? So, if you want to have a holistic tank with a natural ecosystem, you've got snails and little creatures that clean up after your fish, that eat algae, that control the waste. You've got beneficial bacteria keeping your cycles healthy and fixing nutrients and things for your plants. Where do you want to get this stuff from? Should you draw it from an aquarium like this one? This is a sample from an aquarium that has been stable for 5 or 6 years, and this is water from it. Specifically, we took 10 turkey baster samples from down by the substrate and mulm from 3 water sources. And here you can see we've got what was in this fairly stable aquarium. You can see there's some Daphnia and some other little things, but for the most part, it looks like pretty clear water. Then we've got the outdoor ponds where I am raising my Daphnia and other live cultures that I feed my fish and strain out. That's why there's all this algae, is because they eat algae. And then we're going to look at if you went to a local lake and got the leaves and debris from the local lake, what else would you be bringing back? Are there good things like worms and beetles and insects that are helpful in our aquarium, or are these going to end up being a pest in our aquarium? So we're going to look under the microscope to get an idea of what's in even the clearest of water, and we're also going to observe these containers for a number of days and see what develops. So, which is the best way to spark a naturally diverse, stable, and healthy ecosystem? We'll find out. Hello, my friends. I'm Alexander Williamson, and welcome to FISHTORY. Microbiodiversity. Where does it come from? When does it appear? And so when you have an aquarium, a lot of people right now, myself included, are very interested in keeping some level of microorganisms around for your fish to eat, to break down waste, to convert nitrogen, to do all sorts of different tasks in the aquarium. And this diversity is found in nature, right? So is it faster to jumpstart the aquarium drawing directly from nature, or is it quicker to use an already established aquarium because that's already the same temperature, it's already the same like bioload of fish and plants, substrates and things like that? Since it's such a similar condition, is that going to be a better way to do it? Or is there another way where we can take buckets outside and let those age, and them being small, they're not going to have the big bugs and beetles and fish and things like that, but they will allow us to draw those micronutrients, those little types of water striders, fleas, scuds, things like that, that our fish will eat, and also bacteria, fungi, archaea that our aquariums may use. So the question is, when we go out and sample in these bottles, I've got 3 of these bottles, we're going to take one from a 1-year-old tank here, or tub. We're going to take one from the 5-year-old tank in my house, and we're going to take another from the pond down the street, and we're going to see what we find under the microscope. And I'm curious to see, what's going to be useful. If it was all dragonfly larvae and things like that, that's not very useful because they're so big that they're just going to harm nanofish. Whereas if we get a whole host of little tiny microcrustaceans and little invertebrates and things like that, then we might be talking about some really interesting results. And maybe it's better to go get it from the wild directly. Maybe it's better to make sure that you're kind of keeping an eye on it and you know what's in it, like in my tubs. Or maybe it's best to have total control and be really observant in the aquarium. Not sure. So let's take a look under the microscope and see which one is providing the most feeding options and the most microbiodiversity out of the 3. All right, my friends, we have our, to most people, pretty gross pond water here. Lots of mulm in the bottom, lots of stuff that's breaking down. So I'm going to be curious. We're going to test to make sure that their nitrites aren't too high. Also, quite a bit of algae growing in here, as well as it looks like some cyanobacteria, and there are quite a few little critters moving. Now, I filled this up using 10 squirts of a turkey baster, and I figured that this would limit how large of insects we would get or anything that's just distracting to us right now. I want to know about the microflora and fauna that's going to help us cycle the tank, break down waste, and provide food for our critters. Here we are at the little park where we're gonna get the water samples, but I wanted to show you guys it's sunset and there are baby bunnies all over. All right, so here we are at the little local pond. You can see, not a huge pond. It's real shallow. I think maybe 5-6 feet deep most of the way, and you know, only a few inches. Hey, look, a little rat, or is it a muskrat or a rat nutrient? It's a rat. So we got a little rat there in the city, and you can see all the little circles forming. That's either little insects or flies, or it's fish. There's actually quite a few bluegill and carp and small fish in here. But you can also see that the algae is very thick. So let's see what we get from the shores of kind of a stagnant lake or pond. You can see in some spots where there's bluegill nests that have been cleared out. So we'll see what we can get. All right, so you can already see that we are getting a lot more stick debris, algae, and leaves. I'm gonna try to shove some of that in here just because there is so much of it. All right, so I want to show you guys what is actually here, and I'm not getting it with the turkey baster. We're not getting a full view of some of the stuff that's here. So I just threw out a scoop, and before it gets dark, I want to show you guys what's just in an average leaf litter here. So you can get an idea. We don't need a microscope for this stuff. This is all potential food for fish. You can see here's Gomeris, scuds. You can see that there are worms, there are oarsman beetles, or definitely a lot of mosquito larvae here. There are water boatmen, there are back swimmers. So here is a little fish just from the side of the water. I was able to scoop it up, and you can see how much algae is here. They're hunting for all the little insects in the algae. Alrighty, my friends, this is the tank that we are gonna pull from to sample what is going on in a planted nature aquarium. This tank is going on 6 years, and these little hovering zebra loaches are currently the ones spawning in here mostly. Also, I do have some red arc pencilfish that are doing well in here, and there's a whole lot of Saswassertang in here, and there are also lots of different layers of substrate. So we're going to take a little bit from the mulm down here at the bottom, and we're going to also take from the water column and then kick up whatever's in here in bits of algae and Saswassertang, and we'll take a look at that underneath the microscope. But it's a very healthy, stable tank that is rather productive. So I think it will be a good model and a good example for what a planted tank can be. All right, my friends, it's been a very hot day, and finally it is nighttime and things are cooling down. But in these containers, what we've had happen is all the little worms and critters have come to this cooler side of the bottle that I had facing the air conditioner over there for the last few hours. Also, it's been dark over here, and so now we've got our aquarium sample, our pond or tub sample, and our lake or large pond, whatever you want to call it, sample here. What you can already tell is that there are a whole heck of a lot of these little flatworms and worms, detritus worms, planaria, and scuds in this sample. Now, a lot of people ask me, Alex, where did you get your scuds for feeding your fish? Because they know that I raise scuds intentionally in an aquarium of their own. Well, I went and I got them out in nature, just like this. Some of them also came from the hobby. So this water right here represents stuff that only entered because of the aquarium hobby, both on plants, on fish, or perhaps on decor or substrate. But in 5 or 6 years, you move things in and out of your aquarium. Things also come in on your clothing. So whether it's little spores or little eggs that are hiding, they do eventually make their way into your aquarium, just like the beneficial bacteria magically appears in our aquariums, as do fungi, algae, and all sorts of other things. And it's very controlled when it's through other organisms like fish, shrimp, and plants or snails that could have a couple hitchhikers or eggs or something like that, versus being completely open to the elements, obviously. let's take a look under the microscope. But I think I've seen what I need to see just by looking at these 3 samples. I just feel that there are so many different things going on here, so many potential micro habitats within that singular pond even, that I don't know that I would want to add even just leaves from this ecosystem and trust that it's going to be safe for my fish or shrimp. There are so many dangerous little parasites and things out there, even if a lot of them are going to be eaten by other little critters out there. But I mean, look at this little ball of scuds. Right next to it, we've got a hydra. You can see that looks like a little sea anemone there. And that, I've shown in other videos, is able to take out shrimp and baby fish, no problem. There's another hydra right there and another hydra right there. You can see these green hydras on the wall. Also, just the massive amount of planaria, which can sting and bite and kill your shrimp as well. And here we can see there are definitely many species of them in here as well. And same with the scuds, many species. So maybe we could take some of the scuds out of here, culture them, and see how they do. But here you can see there's a little detritus worm right here that's much skinnier than the planaria above it. And maybe we want that detritus worm to help break down our mulm and substrate. But at the same time, do we know that that little worm is not something like a tapeworm or a pinworm that's just going to burrow into our fish and cause problems? You can make your own decision, and I'd like to hear your opinions, but I'm just here to show you what's going on at each of these different options that you might want to start your tank with. The problem I see is there enough diversity in here, in this mostly clear looking water, to even feed your fish? I'm trusting that the bacteria and the fungi and the archaea, all the stuff we can't see, it's too small, I do trust that that's in here and that it's on surface areas in the aquarium or on the mulm, on the debris in here. I know that from letting it grow out on agar, on petri dishes as well. But is there enough life in here to really give your fish food and also your ecosystem stability and diversity? Let's investigate. Let's look under the microscope. These critters are far too big for the microscope, so we'll be looking mostly under here. And I will take a look under the microscope here to make sure we're not missing anything major, but I have a feeling it's going to be a lot of odd little circular creatures that we can't identify. And I'm curious where this sample will stand, if it will be somewhere in between, or if it's going to be a lot cleaner, or if it's going to be as dirty as the lake. So let's find out and finish up our little investigation. All right, so right off the bat, we're going to look at 100 times zoom of my aquarium. Now, there's some debris, but mostly this is clear looking water. I wanted to get water with just a couple little flecks of stuff in it, because we could see with my phone zoomed in, and we spent plenty of time in the video looking at the bottles, at 20 times zoom or 50 times zoom, but I want to see what's going on really close. And here you can see there's fungi, there are fragments of plants, there is mulm, which is covered in bacteria. You can see cyanobacteria fibers down below and algae fibers as well. The fungal fibers are in there too. So you've got a real diversity going on here, which is pretty interesting. And there are little creatures. There's also little cilia or some type of hair, maybe from a plant. I can't tell for sure, the clear one. But as we look through here and look around, that debris all has beneficial bacteria. But what we're not seeing are a ton of these little single cell critters like that little floaty orb, which so far I can already skip ahead and tell you there are a lot of those in the lake sample. Now here we have a little paramecium or something along those lines. These are what newborn fish are going to like eating, and what you can create by taking a piece of lettuce or broccoli or sawassertang, any type of, dead leaf material, and you just throw it in the water and let it sit in the sunlight. And eventually these things will kind of sprout forth. They're living organisms on there. And Zoomed in here, you can actually see the little cell walls. And that's why they're called cells, because they look like little cubicles from a monastery, and monks discovered them looking closely. So we're going to zoom in more, and here you can see there's a detritus worm wedged in there and eating some of the sawassertang. And now we're going to skip over to the lake. And at the lake, you can see there's debris and diversity, probably more diversity. Also, there's more live algae. There goes a scud. So there's a scud. Now, keep in mind that this is giant for the magnifying glass. This is big enough to see with the naked eye. Now, these other little things, the little seed shrimp or ostrocods, those are not really big enough to see with the eye. I mean, they just look like little dots. But here we're seeing a lot of cyanobacteria that's alive and well. So if you put that in your aquarium, I don't know, it might start actual blooms. Also, here we can see great food sources. But again, I like to capture them, and I was hoping they wouldn't even be in this sample. They're all small enough. They're babies. And there's another little seed shrimp. They're all babies because, I wanted the water that was clear. We could already see what was going on with the organisms big enough for our eye. But what's really interesting here is that you get a real diversity, even at the small level, of things like this little mite, where we could actually make out its heartbeat. And there goes a paramecium right in the corner too. Now, you can see that, like, we don't know, are these harmful? Are they helpful? Are they just protein? Here's a little something in a shell of some sort. And again, there goes the little parameciums, and there is a hydra. Now, what's interesting about seeing this hydra is these green hydras in particular actually have hundreds of little algal cells in them, and they are a photosynthetic life form. And that's actually a very new discovery, that they've kind of discovered how that works. But they are able to use the energy and share it with the algae, with the chlorella algae that's inside of them. But they are still stinging and still considered pests. But they are an indicator of good water quality, which is interesting, seeing where that lake was. Okay, so when we're looking here at the tub, you can see we got seed shrimp, and we've got, I only see 2 types of algae and no real cyanobacteria, but yet again, we do have a little spider mite or some sort of mite. So I think those bigger ones might have been duck mite or even fish mites, whereas here we've got something really small, really small. And this water doesn't have a lot of the other floaty stuff in it like the other samples did, because it's a year-old tub. And so I guess it makes sense that the diversity is a little less. We'd have to guess at the fungal and bacterial diversity, but here you can see there are organisms, even really small single cells, coming and going from the little ostracod and the little mite there. So it all kind of adds up the way I thought it would, but I want to show you something. All right, so I want to unhook here from the microscope, and we'll come down and look at the slides that we have been looking at. Now, that whole leaf section of the sawassertang that we saw under the slide, that's this entire piece right here. And we saw all those thousands of cells that make that up. And another way to get an idea of what we're working with, right here, that the biggest scud we saw is right here. That is its body right there. It is the size of the ballpoint pen. So these are not the scuds, this is the size of my hand. These are not the scuds that you see in the samples, that are big swimming around like these guys here. These are all the stuff we saw is so small that it looks just like floating debris at the biggest. So I just want you guys to get a sense for that's what I was interested in under the microscope, is what's going on in these setups. So in the lake water right here, and look at all those worms and things that fall down, all of those creepy crawlies and life forms and protein, good or bad, but look at it all. That's all too big for the microscope. Whereas the tubs we had, there's a little bit of those life forms, but it's pretty much the stuff I've selected, and a few insects and their larvae. And then in the aquarium, it's basically very, very, very small organisms and a few pests, scuds, planaria, and a hydra or two, compared to all the hydra that I keep finding in here from the lake. It's really mind-blowing how many creatures were living in this sample. All right, my friends, so what is my consensus after several days of studying these different bottles? So it's been 4 days, we've had these bottles, and time has passed. I mean, there are mosquitoes that have hatched in these bottles, for instance. So you can find out what is going to be, that's from my tubs, what's going to be in there. Also, little snails have hatched in these containers. And in the case of the lake water, absolutely all sorts of little planaria and worms and flatworms and non-flatworms, segmented worms, tapeworms, pinworm type things. They've all come out. And just by watching these containers, I've also seen that the snails have these kind of corkscrew-like worms that are coming and going from up in their shell area. And sometimes you can even see them coiled up in the clear recesses of the various organisms that are here. Now, granted, all these things have a place in the wild, and they are going to be in there to break down, things that are decaying and things that need to be broken down. But on the scale of an entire lake or a large pond, or for that matter, that whole drainage basin that empties into whatever body of water it is you're working with, you're working with very different organisms, and they are needed to break down an entire forest full of trees and leaves that maybe shed into the water every year, and the type of algae that builds up, and salmon or whatever it may be, bass or perch that are dying in there, and just all the things that nature produces in a year. That's not needed in your aquarium, though. And so I like to have some control. I like to specifically look for an organism I want, like a scud, like this little scud here. Try to identify the species and the genus, the genus if I can, and then if I can sort that out, and I know the species as well, that's even better. And then, what I can see here and what's kind of interesting and hopefully educational is that when we looked under the microscope, regardless of if the water was bright green or if the water was a dark green or if it was just pretty clear with little bits of debris in it, this 5-year-old tank, that debris, when we zoom in and look closely, it's all living organisms. And it's tiny ones. And some of them may be parasitic. They may be feasting on one another. There may be some little pests. Like, for instance, you wouldn't want these scuds in a tank that was for shrimp, so I wouldn't use anything where there's scuds to jumpstart a shrimp tank. But if I'm doing a holistic ecosystem, and I know that the shrimp are going to possibly not survive, and that maybe only some of the shrimp will survive, and I want snails and I want all sorts of critters, then I could definitely still stomach using something like a pond that is essentially been curated by me. Only the flying insects and some random traveling, critters end up in here. So I know very well what is in this for the most part. And at a microscopic level, we saw that the old tank had almost as much diversity as the wild lake, which to me is absolutely wild. But even just giving it a couple days without fish around, this bottle is just springing forth with life, whether that is little seed shrimp or little Daphnia. All of that was hiding and waiting to be born and to just hop out. And you don't really need a microscope. You guys can do this at home with your camera and spot a lot of the things. I mean, there's a little hydra back there. There's some Daphnia here, young Daphnia. And there's also paramecium, those little rod-shaped guys. There's stuff in here, but I know that this tank has nothing that is super harmful. I haven't had to medicate it in a long time. It's been stable and trustworthy. And so for me, I would feel happy to jumpstart a tank with this. I would just be aware to not put scuds in, probably, because that's something I want to control later once the fish are in it. I don't know if I would even use the tub to jumpstart my tanks anymore, quite frankly. I would if it's a nature tank with mid-sized fish and things, but if I'm doing the really tiny nano species, there's just so many things that could meander in here, especially if I lived in a tropical location. And the fact that, mosquitoes are in here and bloodworms are in here, I think we can still target those things and selectively feed those to our fish without having to add all the other random mystery that's in here, let alone adding something like this. So that's my takeaway. Is my first preference is to use the aquarium water. It is full of life. It will keep your nanofish alive and eating snacks no problem for a long time. Plus, it has all the diversity of the algae without any one taking over, It has so many competing that they're keeping each other in check. Whereas over here, you get big blooms in one section of cyano, another one of one type of algae, another of another type of algae, and then clear water, which isn't really clear water. There's actually algae and bacteria and all sorts of stuff in there. And here you get kind of a monoculture of algae, and it's keeping the critters that like algae alive and the insects that are in their larval state alive, and snails and things like that. But I use this as my reservoir to selectively then strain and siphon off what I want to then add to the ecosystem that is my tank. And ultimately, I get the most control with that. And that's what I think is probably the most sensible way to approach this. But if you disagree, I'd love to hear from you. I think that there is value definitely in using completely wild stuff and letting it go wild and see what happens. However, there's just too many variables for me to understand all that's going on and to try to care for my fish when there's that many organisms in play and that many processes and, decay and deposition and different trophic levels. It's a lot. Managing the planaria and the scuds is enough work for me with this system. And that's kind of where I stand. And also, it's nice to get the insects to hatch first, so you know what you're dealing with, so you don't end up with mosquitoes flying around your house and whatnot. All right, my friends, we have come to the end of another episode, and I really want to thank you for letting me do all of this kooky stuff and collect, slides of different critters. I collected over 30 total and looked at these 3 different bottles of water, let them mature, and it takes time to make videos like this. So hopefully you guys gained something insightful from this, and you don't have to take the time to do it. That's part of the goal, and to just answer those curiosities that maybe we all have. And maybe there's an answer to a question you didn't know you had yet in this video. And when you come to that issue, you'll say, you know what? I remembered this one thing in that video, and it helps me solve, yes, a worm can be living inside of a snail as a parasite, or whatever may be the solution. But that's why I like doing videos where we get to do a deep dive. So I really want to thank you, because you allow me to do that by watching, by subscribing, by becoming members, super chatting, and, just by being supportive, by commenting. All of that is so incredible, and I just can't do it without you. So I want to thank you again and again. I hope that you guys all have a great day, and that there was something interesting in this episode that excites you. Let me know what it was in the comments, if anything really stuck with you, because maybe I'm missing something. Maybe there's some revelations that I came to incorrectly, and so I like to hear lots of opinions. Regardless, I'll keep at it if you keep coming back, and I'll see you next time on FISHTORY.